Hello there, and great day to each and every one of you, my class. And today we will continue to our quarter two. So, congratulations, you passed our quarter number one. So, to start with, thank you very much for the leading of our opening prayer for today. And the secretary kindly check the attendance. How many boys and how many girls do we have right now? Don't forget to send the number or the total number of boys and the total number of girls in our group chat. And for our quarter two, week one, we are going to tackle about microscopy. So microscopy simply teach us in this module the following lessons. So specifically, this module will help us to learn about number one, the history of microscope and its uses. Lesson 2, parts and functions of microscope. Lesson 3, basic microscope technique and rules to follow. So let's just have a short looking back on what microscopy is all about. So direction, how the following are being observed. Accomplish the table. So this is just a recap or the prior knowledge that you know about microscope. So we have the following organisms. We have a dog, COVID virus, animal cell, and then plant cell. So siguro yung iba sa inyo, you are familiar with those terms. And now, we are going to identify what do we use to observe them. By naked eye, by magnifying glass, or by microscope. So for example, for number one, pet dog. You can observe this by what? Via naked eye, magnifying glass, or microscope. Okay, the correct answer is by naked eye. So meaning to say, you just need to use your eyes for you to see your pet dog. You don't need magnifying glass. You don't need microscope for you to see your pet dog. How about COVID virus? Okay, so you can see COVID virus via microscope or with the use of microscope. And then animal cell. So we are talking about cell, not just the animal. Okay. The correct answer is also microscope. And plant cells. Okay, very good. So you can also observe plant cell in a microscope. So meaning to say, we can say that microscope is used for us to see very minute objects. So when we are talking about minute objects, we are talking about very small objects. Okay, so that's how are we going to use the microscope. So for lesson number one, we are going to tackle about the history of microscope and its uses. Are you ready to listen? Okay, let's proceed. So as an introduction, the microscope is a tool that can help you to see tiny objects and organisms. It makes objects look bigger because of its magnifying power, or what we call magnification. So meaning to say, when we are talking about magnification class, napapalaki nito yung bagay na maliliit. Okay? It is also help us to see the capacity to distinguish small gaps between 2 points which we cannot distinguish through its resolving power or resolution. So pwede natin makita dito, for example, yung nucleus ng cell, yung cell membrane ng cell, and then yung cytoplasm ng cell which we cannot see, okay, without the use of microscope. Pero kapag gamit natin ng microscope, kitang-kita natin yung mga bagay na yon. And then during the 1st century Romans, they discovered a clear thick glass with thin edges. They found out that if you look through an object using this glass, the object would look larger. This is where the discovery of lenses started. So it is just like accidentally discovered when Romans saw a glass that can make the object bigger. Parang mumukhang malaki gamit yung glass na to. So Basically, that is when the magnifying glass started to be used. Okay. So these are the examples of the earliest, okay, microscope that they use using a simple magnifying lens. And it all started in a magnifying glass. So the year is in the 1st century AD, year 100. The inventor are the Romans. So this is what it looks like. Siguro nakita na tayo ng magnifying glass, di ba? So what are the features of it? So they were able to discover a clear glass that was thick in the middle and thin on the edges, and that if you look through an object using this glass, the object or the thing that you are going to observe looks bigger. So the same lang din ang ginagamit natin sa mga salamin or reading glasses na meron tayo. Diba, yung gamit nating reading glasses, kung kayo ay nakasalamin, it looks a simple glass, but you can see that this glass can make small objects looks bigger. For example, nagbabasa kayo ng mga textbooks na maliliit yung mga texts. Okay, yung mga writings. So, this magnifying glass or your reading glass will make those small objects looks bigger. Okay, so dun nila nagsimulan kunin yung konsepto ng microscope. And then the next one, we have the first compound microscope in 1590 by Zacharias Janssen and his father Hans. And this is what it looks like. Okay, the first compound microscope is like a hollow tube. And then they took a tube and put lenses on each end and observed objects. He made a very important discovery of the first compound microscope. It's like, siguro kung nakakita na kayo ng kaleidoscope, para siyang tube lang na nilagyan nila ng lens. And then, they use it to observe things. So, they put it in a tube. And then, we have the discovery of Anton van Leeuwenhoek in 1632 to 1723, and this is what Anton van Leeuwenhoek discovered. Okay, so it has a simple holder, lens, a knob, so that you can adjust it. Okay, pwede mong i-adjust yung itsura ng object. It's either you wanted to see clearly or you wanted to see it afar or near, something like that. So this is what the microscope of Van Leeuwenhoek looks like. He is known as the father of microscopy cells. He made a simple microscope and was the first to observe bacteria, yeast, red blood cells, and describe colonies of protozoans teeming in a drop of rainwater, which he called animalcules. Okay, so before the word microorganisms is not yet postulated, so they call it animalcules because when he saw the protozoans, it's like an animal, it's moving, a cell that is moving. And then we have in 1665 by Robert Hooke, it was discovered by Robert Hooke, and this is what the microscope or compound microscope looks like. It has a stand already in a tube. So he spent improving the design and capabilities of the microscope. He discovered tiny compartments in a thin sliced cork. He then called this compartment cells. Okay, so nagkaroon ng improvement pagdating kay Robert Hooke, yung ginawa ni Anton van Leeuwenhoek. Then during 1826, the inventor was Joseph Jackson Lister, and this is what it looks like. I want you to remember the picture, and it is displayed in the Museum of the History of Science. Okay, he developed an achromatic lens and induced several weak lenses together at a given distances, resulting in a great magnification without blurring images. So makikita mo agad ng malinaw. This helped makes microscopes important tools in medical research. So dito na nagagamit yung microscope when it comes to the medical field. Kasi mas clear na yung nakikita nilang, for example, bacterial infection. So they can able to identify kung anong bacteria yung naka-infect dun sa tao. And then during 1931, it was invented by the physicist named Ernest Ruska and electrical engineer Max Knoll. It's a bigger version of a compound microscope. He developed the first prototype electron microscope, and it's capable of 400 power magnification. It's a very big microscope so that the magnification is very big. Okay, so dito na nagsimula yung mga electronic microscope. Okay, so as time goes by, there's a lot of microscope right now. Kung napanood niyo yung introduction video natin, nandun yung example ng mga microscopes right now. So for lesson number 2, for the function or the parts and functions of the microscope, basically we are going to focus on the compound microscopes that available in our school, and also that can be used with your own hands without the use of electricity. Okay, so let's start. So parts and functions of the microscope is divided into 3 main parts. We have the mechanical part, magnifying part, and then illuminating part. When we are talking about mechanical part, these are the parts used to support and adjust different areas of the microscope. Okay, using your hands basically. So gamit yung kamay nyo, pwede nyo siyang i-manipulate. Okay? And then we have the magnifying part. So these are the lenses used to enlarge the specimen or the objects. And then illuminating part used to provide light. Okay? Because you cannot see or hindi mo kasi makikita yung object na yon under the microscope without the presence of light. Kapag gamit mo ay electronic microscope, basically may bulb. Kapag hindi naman, yung simple microscope lang, simple compound microscope, you just need natural light. So, makikita mo doon yung mirror. Do not forget the 3 main parts of the microscope: mechanical, magnifying, and illuminating part. And for us to start with the different specific parts of the microscope, we have letter A, eyepiece. And this is where the eyepiece is located. It contains the magnifying lens. It is where we look through. So, dyan natin ilalagay yung eyes natin for us to see the specimen or the object. And while you are looking at the eyepiece, be sure that both eyes are opened. Do not close your one eye for you to see. Okay? Because there's something wrong with your vision if you close your one eye. Maybe you can put your hand here, or which eye you wanted to put your hand, as long as both eyes are open. Okay? So, dapat ang parehas mong mata nakabukas. Kahit yung isa nakatakip nakabukas na, huwag kang pipikit na ganyan kasi lumalabo. Diba? Try nyo. And then, letter B, we have the body tube. So, this is the body tube. It holds the eyepiece and lenses in position. Nandyan sa body tube yung mga lenses. Meron kasing lens dito, may eyepiece, at may iba pa tayong lenses dito. At dyan siya situated. We have the coarse adjustment. Ito yung biggest knob. The large knob which moves the body tube to produce large changes in focus. So, pwede mong i-adjust yan mas mapalapit dun sa specimen or mas mapalayo sa specimen. So that you can also adjust the clearness of the object or the specimen. So, pwede siyang maging malinaw, pwede siyang maging malabo. And then, for example, tapos na natin i-adjust yung coarse adjustment. We have fine adjustment. So, after you adjust the coarse adjustment, so, sa tingin nyo malabo pa, this knob will also moves the body tube to produce small changes in focus. So, for example, na-adjust nyo na yung coarse adjustment, medyo malabo pa rin. Use the fine adjustment. And this is what the fine adjustment looks like. Okay, nasa taas yung coarse adjustment, nasa baba niya yung fine adjustment. And then, we will go to revolving nose piece. And this is what it looks like. Okay, andyan ang revolving nose piece. It is a circular metallic piece holding the magnifying lenses to the tube. It revolves freely so as to select the required lenses needed to focus. So, pwede mong ikutin yan, but gently. Okay? Kasi pipili ka ng lenses na gamit mo. Meron tayong usual na tatlong lenses na gamit. So, pwede mong ikutin yung which lens that you're going to use for you to see the object. And then we have the low power objective lens. So we have three, right? So low power objective lens or LPO, it is the lens with 10 times magnification. Do not remember that. Kasi para makuha mo yung total magnification, kailangan mong i-multiply yung lens magnification ni eyepiece tsaka lens magnification ni LPO para makuha mo yung total magnification. And then we have high power objective lens or the HPO. Usually may color distinction. So minsan kapag LPO may isang line na color blue, kapag naman HPO or high power objective lens may dalawang strip na kulay red. So it depends. So HPO is the lens with 40 times magnification. And then the last lens is the scanning power objective lens with 4 times magnification. So scanning power usually it is the basic, yung pinakamaliit. And then we have the scanner or scanning power. And then we have the low power objectives and then the high power objectives. And then we have stage. So this is what the stage looks like. The stage is the flat surface on which the slides are situated or placed. And then we have stage clips. There you go. Those very thin metallic part. So stage clips simply holds the slides in place para hindi gagalaw-galaw yung slides mo. But there are some compound microscope na may mga knob na pwede nyo rin siyang i-manipulate para magalaw yung slides. Okay? And then we have letter K, the iris diaphragm. So iris diaphragm is located at the bottom of the stage. So it functions as a regulator of the amount of light entering the body tube. So dito mo pwedeng kontrolin yung ilaw na pumapasok doon sa microscope natin, doon sa iris diaphragm. So next is the base. The base provides support for the entire microscope. So importante yung base na yan. Kasi dyan yung foundation kung saan it supports the entire microscope. And then we have a mirror. Usually we use convex mirror. Okay, and this is what convex mirror looks like. But here in our example, they uses electricity. So may bulb dyan. In mirror, while we are using this mirror, it collects light from the surrounding and allows to reflect upward. So kaya ginagamit natin yung convex mirror. Mas madaling mag-capture ng light. Okay, naka-convex siya. Okay, and concave. So it depends kung anong gagamitin nyo doon. Meron tayong concave mirror, meron tayong convex mirror. And then the arm. So this letter C structure of the microscope is the arm. It supports the body tube. So you can also use to hold the microscope in the arm and also at the base for its proper carrying. And then we have the inclination joint. Inclination joint is this small circle at the bottom of the arm. It makes the microscope to tilt the arm back and forth. So you can tilt it back and forth. For example, you are looking for light kasi medyo madilim yung kwarto, naghahanap ka ng ilaw ngayon, so pwede mong i-tilt yung microscope mo, and that's the use of the inclination joint. And for lesson number 3, the basic microscope techniques and rules to follow. So ito yung mga dapat yung tandaan. Number 1, microscope care. So remember to always carry with 2 hands. One at the arm and the other one at the base. This is the proper way for you to hold it. And then only use lens paper for cleaning. If you have the microscope kit or cleaning kit, use it. And then do not force knobs. As much as possible, use it with gentleness. Always store covered. So kapag binalik niyo siya, kung meron siyang cover, ibalik niyo siya dun sa cover niya. Kasi napaka-sensitive ng mga lenses. Okay? And then we have keep objects clear or your workplace clear of desks and cords. So if you are using the electron microscope, be sure that you unplug it, and then as much as possible, please keep the wire safe. Okay, huwag yung pakalat-kalat. So this is how you hold it first, and use the cleaning material or cleaning kit with the microscope, and then please do not make use of the knob with force. Okay, and lastly, please make your working place clean and neat. And for the steps on how to use the microscope, remember this so that when we are in the face-to-face in our classroom or laboratory, you will not forget the following. So number one, if you have the specimen already, mount the specimen on the stage. So that's the first thing. If you have the prepared slide or prepared specimen, you can do it. Okay? Then number two, optimize the lighting. Look for the light. Adjust the condenser, the iris diaphragm, and then think about what you are looking for. So if you are looking, for example, you are looking at a, we can say a cell, specifically an animal cell. So you are looking for the nucleus, hinahanap mo yung nucleus o kaya yung cytoplasm gamit ang microscope mo, then you can adjust the stage clips. Okay, so meron doon. Pwede namang gamitan mo na lang ng kamay or yung sinasabi kong knob, stage clip knob. Then focus, locate, and center the specimen. Adjust eyepiece separation to focus. Then select an objective lens for viewing. Then at last, adjust illumination for the selected objective lens so that you are good to go and you will be able to see the magnified objects or magnified specimen. And do not forget to answer the following for our quarter 2 week number 1. Watch the video to review, and you can also rewatch and answer all the questions in your modules in your Google Classroom. So you will see the module in your Google Classroom, and please follow the instructions. So if you have your soft copy, you can answer it in your notebook, but if you have your printed modules, you can answer it directly there. But do not forget to send it or turn it in our Google Classroom. So for lesson number 1, pages number 3, that's the pre-test, you will answer that. Then pages 5 to 6. Again, pre-test first and then pages 5 to 6. For lesson number 2, answer pages 6 to 8. And for lesson number 3, answer pages 8 to 10. And do not forget the last page, post-test, on page number 10. Okay, do not forget all these pages for you to complete your task or your written works for week number 1 of quarter 2. So if you have any questions or clarifications regarding our topic for today, you can reach me through my different social media accounts. And please focus on our group chat. I will announce there and give you the reminders. So please open your GC and learn how to backread. Okay? Then that's the end of our quarter 2 week 1. I hope you will have a very nice day, and please be excited and let us learn more about biology.